So now let's look further, and this time look at cell structure. And further information, as always, you could find them in the individual articles on Wikipedia. So in this case, animal cell and plant cell. So every cell is enclosed within a cell membrane that separates its cytoplasm, which is all the material inside a eukaryotic cell, except the cell nucleus, from the extracellular space, which is outside of the cells. A cell membrane consists of a lipid bilayer, so 2 layers of lipids, as I showed earlier, including cholesterols, which is a type of lipid that sit between phospholipids to maintain their fluidity at various temperatures. A number of phospholipids have that hydrophilic end with 2 hydrophobic tails. So the top end likes water, the bottom tails don't like water. So cell membranes are semi-permeable, so allows some stuff but not other stuff, allowing small molecules such as oxygen, carbon dioxide, and water to pass through while restricting the movement of larger molecules and charged particles such as ions. And cell membranes also contains, I think that's a typo, should have been just contains, not the S. But I copied and pasted exactly as is from Wikipedia, so I just added this SIC. This is the Latin phrase sic. And then I actually added it just now to the inline commentary. So if I just search here, scroll all the way back up here, so do a search, and I should have, actually this is the beginning. Yeah, so I just added it now. I thought it was, should add it. I just had it inserted, but I didn't add it here. So go back to the inline commentary. So SIC with the brackets and bold. This indicates quote is copied as is, which includes any errors or spelling mistakes. And it's from Latin sic erat scriptum, meaning that thus was it written. So yes, fascinating stuff. Let's go back over here. Yes. So let's read that again. So cell membranes also contain membrane proteins, including integral membrane proteins that go across the membrane, serving as membrane transporters, and peripheral proteins that loosely attach to the outer side of the cell membrane, acting as enzymes shaping the cell. Cell membranes are involved in various cellular processes such as cell adhesion, which is interact and attach neighboring cells, and storing electrical energy by maintaining a voltage or electric charge difference between the inside and outside of a cell. And cell signaling. So just remove this A, just so inside and outside of cell. In other words, whichever one the membrane is on. Yeah, or put the cell inside and outside of the cell. And cell signaling, which is the ability to receive, process, and transmit signals with its environment and with itself, and serve as the attachment surface for several extracellular structures, such as a cell wall, which surrounds the cell membrane of some cells. And then you also have, another extracellular structure is the glycocalyx. Yeah, so I believe it's pronounced glycocalyx or glycocalyx, either or. So which this one is a generally fuzzy hair-like attachment surrounding some cells, and cytoskeleton, which provides shape and mechanical resistance to deformation within and surrounding a cell. So yes, a lot of technical terms in here. So going further, so here's a general structure of an animal cell depicting various organelles, which are subunit within a cell, analogous to organs in a body. So here is just a general overview of the different stuff inside. Now let's look at each one of these and also some electron microscope images of them. So a diagram of a typical animal cell, organelles are labeled as follows. So nucleolus, I think it's pronounced nucleolus or nucleus, or nucleus. Let's try that again. So nucleolus. This is the largest structure in the nucleus, and it produces ribosomes. So let's go. This is the number 1. This is the inside the nucleus. So this number 2 should be the nucleus then. It's going over that soon. So there's the nucleolus, and then there's the nucleus is the around. This is the largest structure inside, and it produces ribosomes, which I'll get to in a bit. And then there's the cytoplasm, which is everything but the nucleus that's outside. So electron micrograph of a HeLa cell. And a HeLa cell is an immortal cell line used in scientific research. It is the oldest and most commonly used human cell line. The line is named after and derived from cervical cancer cells taken on February 8th, 1951 from Henrietta Lacks, which is a 31-year-old African-American mother of 5 who died of cancer on October 4th, 1951. The cell line was found to be remarkably durable and prolific, which allows it to be used extensively in scientific study. The cells from Lack's cancerous cervical tumor were taken without her knowledge or consent, which was common practice at the time. Cell biologist George Otto Guy found that they could be kept alive and developed a cell line. Previously, cells cultured from other human cells would only survive for a few days. Cells from Lack's tumor behaved differently. Yeah, and an immortalized cell line is a population of cells from a multicellular organism which would normally not proliferate indefinitely, but due to mutation, which is the alteration of a nucleotide sequence of a genome, have evaded normal cellular I think it's pronounced senescence. Yes, so I have to Google search the pronunciation. So cellular senescence. Yeah, let's try it one more time. Cellular senescence, which is the cessation of cell division, and instead can keep undergoing division. The cells can therefore be grown for prolonged periods in vitro or in the lab, whereas in vivo is in a living organism. The mutations required for immortality can occur naturally or be intentionally induced for experimental purposes. So that's some fascinating little sidetrack over here on this HeLa cell. So a nucleus, which is number 2, let's go back to it, which includes the nucleolus. Yes, a nucleosis. Or the nucleolus, yeah. So there's the nucleus is number 2. That includes inside the nucleolus. Nucleus. Let's try one more time. Nucleolus, yeah. Yes, pronunciation is not my forte. So so the nucleus contains all the cell's genome, all of the cell's genome except for mitochondrial DNA and plastid DNA in plants. All right, let's continue further here. So here's an electron micrograph of a cell nucleus showing the darkly stained nucleolus inside there. So yeah, I think I pronounced that correct first time. So it's very fascinating. So a plastid is a membrane-bound organelle found in the cells of plants and algae and some other eukaryotic organisms. Yes. Continue further. Yeah, so organelles would be inside the cell. Yeah, so a ribosomes, number 3, it's found floating in the cytoplasm or attached to the rough endoplasmic reticulum, which are dots as part of 5, and synthesize a protein from mRNA or messenger RNA. Let's go look at these dots. So this one is over here. So these dots on there, or even possibly free floating, is number 3 over there. And then, yeah, so those are ribosomes on the endoplasmic reticulum, which is the number 5, or should be number 5. Let's continue further, and yeah, here's a figure of this in action, or just a figure of it. So here's the ribosomes, which are these dots right there, and mRNA supposedly is this thread inside, and then you have these polypeptide chains, or the proteins being created from these dots there. So yeah, it's very fascinating stuff there. And I got this image from here. This is bioinfopakistan.ukmucoz.com. Very interesting stuff. So now here's another image of it. So here's a figure 1 electron microscopy image of simultaneous transcription, which is copying segment of DNA into RNA, and translation, which is synthesizing mRNA into specific amino acid chain to make proteins or polypeptide chains. Yeah, the image shows bacterial DNA and its associated mRNA transcripts, each of which is occupied by ribosomes. And this image was from this, I think it was a textbook or a paper adapted from O.L. Miller et al. Science, this one here in 1970. And this figure is from this website, books.bionumbers.org. And so here's a transcription. This is getting from copying segment of DNA into RNA. So yes, so this is supposedly DNA, this strand, and then it has a strand over here. This is the RNA, or the messenger RNA, and then the ribosomes. Yeah, those are the ribosomes. And they're going to be producing peptides out of this side over there, which is a very, very fascinating look inside if this is actually what's happening. Because the electron microscope images are just, they're not actually moving. They're just basically still images of, like a snapshot in time, and they're also stained. And Yeah, so they're all the molecules are dead. But nonetheless, pretty fascinating stuff. Anyways, let's continue further. And here's another electron micrograph of polysomes, or micrograph, or scope image of polysomes, which are a group of ribosomes held together with mRNA. Yeah, so these are all these ones here held together, supposedly. So this is the ribosomes. An image courtesy of Alexander Rich. And this is from this other website, bio.libratext.org. It's fascinating stuff. So it's very close up, zooming in on these little dots there. Let's continue further. And here is another figure here. This one is from this side, sciencephoto.com. So rough endoplasmic reticulum with ribosomes on the surface, and is a mammal neuron. It's a brain cell. This is put a dot there, and then CNS, or central nervous system, transmission electron micrograph. Yeah, this neuron is from the central nervous system, and this is a transmission electron micrograph, or electron microscope image, or TEM. We'll possibly get to that later in this video. And yeah, so basically we have the rough endoplasmic reticulum, and there's those dots where the ribosomes are at. So very fascinating stuff. Let's continue further. So a neuron, or nerve cell, is an electrically excitable cell that communicates with other cells via specialized connections called synapses. In biology, the nervous system is a highly complex part of an animal that coordinates its actions and sensory information by transmitting signals to and from different parts of its body, basically information sending back and forth. The central nervous system is part of the nervous system consisting primarily of the brain and spinal cord. So now let's look at number 4 is vesicle, which is a structure consisting of liquid or cytoplasm enclosed by a lipid bilayer. So let's go to this vesicle. So it's just a general organelle. And where is the number 4? So here's number 4. This is one right here. This is a general one. I believe these would be as well. Looks like the same shape. Or just, these are possibly, same or different. But anyway, so it's basically a lipid bilayer, and you have either water or other cytoplasm inside of this, enclosed inside. So let's go further into this. So that's number 4. So just a structure consisting of liquid or cytoplasm enclosed by lipid bilayer. And here is some examples here. So the SARFUS, which is optical microscope imaging technique, image of lipid vesicle. So look at these over here. These look like they could either have water or other stuff inside. Just pretty interesting. So let's continue further. So now let's look at number 5 is the rough endoplasmic reticulum, which has it over here with the ribosomes in there. And let's go over here. So here's another image of it. This is also abbreviated RER, and it synthesizes certain lipids such as cholesterol and contains the ribosome. So here's image of here. So micrograph of rough endoplasmic reticulum network around the nucleus, shown in lower right-hand side of the photo. So this would be the nucleus over here, and there's I believe this would be a nucleolus. And then dark small circles in the network are mitochondria and possibly other organelles. So dark small circles in here, possibly mitochondria. I think these ones are the mitochondria over here. But it could be other stuff as well, because they don't all look the same there. But this lines across there, that's usually how the mitochondria look like. Let's go back up to that image to show it again. So this is the rough endoplasmic reticulum, is this purplish reddish over here. And then it contains the ribosomes. Just fascinating stuff. Let's continue going further. And yeah, so this produces certain lipids such as cholesterol. Continue further, and number 6. This is the Golgi apparatus, or Golgi body. Yeah, I think it's actually pronounced Golgi, yeah. So Golgi apparatus or Golgi body, and again, I have to Google search the pronunciation. Alright, let's make this a bit smaller. So this packages proteins into membrane-bound vesicles inside the cell before they are sent to their destination. So yeah, puts proteins inside membrane-bound vesicles inside before they leave. And let's just go, before we get to that, let's look at that again in this in the overview image. This is number 6 right here, and number 6 looks like over here. So this one right here is the Golgi apparatus. So it has this this shape right like that. Very fascinating, right beside the endoplasmic reticulum in that overview image. Let's go here, and this is a micrograph of Golgi apparatus, visible as a stack of semicircular black rings near the bottom, and numerous circular vesicles circular vesicles can be seen in proximity to the organelle. So right here. So it looks like, yeah, this is semicircular black rings over here, and there's our other close by circles there, or vesicles, circular vesicles around it. Now let's look at the cytoskeleton, which is a complex dynamic network of interlinking protein filaments, which are long chains of protein present in the cytoplasm of all cells. Let's go and scroll all the way up to number 7, which is the cytoskeleton, number 7. And there it is number 7 like that. So this is the cytoskeleton inside there. There's another filament across. There's another one across here and across there. And yeah, it gives it strength and so on. Let's go back to the cytoskeleton. Yeah, so cytoskeleton, complex dynamic network of interlinking protein filaments. So it's basically a bunch of long chains of proteins, and is present in the cytoplasm of all cells. In eukaryotics, it is composed of 3 main components: microfilaments, intermediate filaments, and microtubules. And these are capable of rapid growth or disassembly dependent on the cell's requirements. Yeah, and microfilaments are also called actin filaments or protein filaments in the cytoplasm of eukaryotic cells that form part of the cytoskeleton, which are microfilaments, are usually about 7 nm or nanometers in diameter. A nanometer is about a billionth of a meter. And it's made up of 2 strands of actin, where actin is a family of a globular or spherical or global, global shape multifunctional proteins. There's a family of globular multifunctional proteins that form microfilaments in the cytoskeleton. The thin filaments in muscle fibrils. Oh yeah, I think it's pronounced fibrils. Again, I had to Google search that. So they're rod-like organelle of a muscle cell. Yeah, it can be present as either a free monomer called G-actin, which is for globular, or as part of a linear polymer microfilament called F-actin, which is F for filamentous. Yeah, or pronounce filamentous. Yeah, I think that's correct. Anyways, continue further. So intermediate filaments, or IFS, are cytoskeleton structural components found in the cells of vertebrates and many invertebrates. Intermediate filaments are composed of a family of related proteins sharing common structural and sequence features. The diameter of intermediate filaments are 10 nm, is now commonly compared to actin microfilaments, which are 7 nm, and microtubules, which are 25 nm. So the biggest key thing is the different sizes of these. And this is the different type components of the cytoskeleton. You have small, and then you have long, and then you have medium, and then you have long filaments. And these are in terms of diameter. So yes, this is the big one. I mean, big ones. You have these are the thick ones. Microtubules 25 nm, intermediates 10 nm, and then the microfilaments are 7 nm. So thin thin chains. So microtubules are polymers of tubulin that form part of the cytoskeleton and provide structure and shape to eukaryotic cells. The outer diameter of a microtubule is between 23 and 27 nm, while the inner diameter is between 11 and 15 nm. So it's a tubular shape. So tubulin in molecular biology can refer either to the tubulin protein superfamily of globular proteins or one of the member of proteins of that superfamily. Yeah, so a type of protein. A type of globular protein. And then there's several types. There's alpha and beta tubulins polymerize into microtubules, a major component of the eukaryotic cytoskeleton. So here is a image of this. So this is the eukaryotic cytoskeleton, and the actin filaments are shown in red. So these are red actin filaments inside the cytoskeleton. And microtubules composed of beta tubulin are in green. So there's the green ones over there, all interconnected. And so the blue right here is nucleus. That's a nucleus stained with DAPI, which is a fluorescent stain that binds strongly to adenine, thymine-rich regions in DNA. So it can stick to it and show that blue color. Green is the green is a tubulin, microtubules stained with antibody. This one is a body IPY, FL, goat, anti-mouse, IG, and then G, indirect fluorescent antibody stain. And then we'll get to that in a bit. So red is the F-actin or filamentous actin stained with Texas red X. phalloidin. Well, actually, that's pronounced phalloidin. So basically, a type of stain. So fluorescence is the emission. So this one right here, fluorescence stains. Fluorescence is the emission of light by a substance that has absorbed light or other electromagnetic radiation. And then this one, immunoglobulin G, I lowercase g and uppercase g is a type of antibody representing approximately 75% of serum antibodies in humans. IgG is the most common type of antibody found in blood circulation. And this blood plasma, so this is found in serum right here. Yeah, or actually more specifically, let's just go over serum first, just to go over this. So this is approximately 75% of serum antibodies in humans is this immunoglobulin G or IgG. Serum is the fluid and solute component of blood which does not play a role in clotting. It may be defined as blood plasma without the clotting factors, or as blood with all cells and clotting factors removed. And blood plasma is the yellowish liquid component of blood that holds the blood cells, proteins, and other constituents of whole blood in suspension. It makes up about 55% of the body's total blood volume. Yeah, so basically serum is just blood without the, excluding all the clotting factors. And coagulation, also known as clotting, is a process by which blood changes from a liquid to a gel, forming a blood clot. And the factors are all the components that go into this blood clotting mechanism. And the idea of blood clots is, yes, to stop the bleeding. All right, let's continue further. So now let's look at 8, which is the smooth endoplasmic reticulum as opposed to the rough one. And this is number 8. And let's go up to it. And where number 8 is in the overview diagram. And number 8 is right over here. So there's number 8. So this is the rough one in between, in close to nucleus, and just farther out without the dots. This is the smooth endoplasmic reticulum. So let's go all the way back over to it. And this is going to be right over here. Whoops, yeah, so right over here. So the smooth endoplasmic reticulum, SER, it functions in synthesis of certain lipids such as cholesterol and does not contain ribosomes and produces steroid hormones and also involved in detoxification. So here is the electron micrograph showing smooth ER in the arrow right here, and then in mouse tissue, just a line. In mouse tissue at 110,510 times magnification. That's absurd right there. That's absolutely ridiculous. So yeah, zooming in all the way in, and this is the smooth this is a smooth ER or smooth endoplasmic reticulum. Yeah, so it's just this blob right there. It's very, very fascinating. But again, this doesn't look too much like anything. Anyways, continuing further. So now let's look at mitochondria, and these generate most of the cell supply of adenosine triphosphate or ATP for chemical energy, and has its own genome or mitogenome. And this is number 9. So let's go all the way up to it. Scroll up fast with this right here. This is, yeah, there it is. Yeah, so right here, number 9. Number 9 is right here. So yeah, so these are the zigzag lines across there, and these are the mitochondria, which, yeah, they produce the ATP, so the energy, and has its own genome inside. And let's just go up further, and I believe there was, where was it inside this one, I think? No, not this one. There was one of the images that had that, this one right here. So this one had the mitochondria and the dark circles, those lines across, and these ones here. Yes, let's just go over to it. Number 9. Yeah, so chemical energy, so produces the chemical energy, this ATP. So chemical energy is the energy of chemical substances held in chemical bonds that is released when they undergo a chemical reaction and transform into other substances. So here's a close-up of the mitochondria. And look, this is 50 nm, so 50 and then nm is billionth of a meter. All the way zooming in. So 2 mitochondria from mammalian lung tissue displaying their matrix and membranes as shown by electron microscopy. And this mammalian, this is for mammals, from Latin mamma, meaning breast, are a group of vertebrate animals constituting the class Mammalia and characterized by the presence of mammary glands. Yeah, or I think it's pronounced mammary glands, which in females produce milk for feeding or nursing their young. A neocortex, which is a region of the brain. So mammals have, the females have mammary glands for milk production, and a neocortex, a region of the brain, and then it has fur or hair and 3 middle ear bones, and a mammary gland, or a mammary gland is an exocrine gland in humans and other animals that produces milk to feed young offspring. And a gland is a group of cells in an animal's body that synthesizes substances such as hormones for release into the bloodstream. This is an endocrine gland, or into cavities inside the body or its outer surface. This is called exocrine gland. Yeah, so basically into the blood, endocrine, and then into the cavities of the body or its outer surface, or releasing it outwards, exocrine gland. And now let's look at vacuole. This is number 10, and this is a membrane-bound organelle filled mainly with water, containing inorganic and organic molecules such as enzymes in solution, and certain cases including solids which have been engulfed. So yeah, this is a vacuole with a whole bunch of stuff inside there. And let's just scroll up to number 10 in the overview image. Let's go up fast here. So number 10 should be somewhere, over here is the overview. So this number 10 is just a big vesicle here, but it can engulf a whole bunch of stuff there. Let's go all the way back to it. And this is, we're all the way too far here. Let's go back to number 10. This is number 10. So there's a vacuole. So no, the vacuole's inside. No, this is a whole cell. But notice the vacuole's inside. Yeah, I got that mistake. So there's a nucleus is over here. That should be nucleus. And there's a nucleolus or nucleolus. And there's a vacuoles inside that contain some a bunch of stuff there. So it filled mainly with water containing inorganic and organic molecules such as enzymes, and in certain cases solids which have been engulfed. So it could be a solid inside there. So figure, this is from this website, INTL, and then SIYAVULA.com. This is a micrograph of a plant cell. Can you see the clear white organelles, which are the vacuoles? So this one's here, the clear white ones there. The cytoplasm appears very granule in this image. So yeah, it's very granule. So it looks like it's a bunch of stuff there. Let's continue further. So number 11. Cytosol, which is a crowded solution of many different types of molecules dissolved in water that occupy up to 30% of the cytoplasm by volume. And let's go all the way up to it, back to the 11. Yeah, so there's the 11. So where's the 11? There's the 11. This is just pointing to basically, just basically to nothing there. So basically not any of these stuff there. So that's the 11. Let's go back to it. So where's number 11? To learn more about it. Yeah, so it's a crowded solution of many different types, so it's a whole bunch of stuff dissolved in water, occupies up to 30% of the cytoplasm by volume. So it's part of the cytoplasm. So let's go over this cytosol, and this is from this video here. I just took some screenshots. It's very good. It's a very well done video. It just has it all simply shown. So the protoplasm is everything included in the cell membrane, and then it's divided into the nucleoplasm and cytoplasm. Yeah, so if you have a cell like this is the protoplasm, it's everything. Then you have a nucleoplasm, which is inside the nucleus, and then the cytoplasm outside. So cytoplasm is everything included in the cell membrane except the nucleus, and it contains cytosol and organelles. So basically, a cytosol is everything but the organelles inside the cytoplasm. And cytosol is area of cytoplasm that's not held by organelles. And cytoplasm minus the organelles. Yeah, so basically cytoplasm minus organelles. And here's a big overview. So here's a cell membrane. Everything inside the cell membrane is a protoplasm. Then it's divided into the cytoplasm, which is everything but the nucleus. And in that cytoplasm, it contains all the organelles and the stuff not in the organelles, the cytosol. And then nucleoplasm is inside the nucleus. And yeah, so that's the nucleus and everything inside. So there's the nuclear membrane and everything inside it. So yeah, it's very, very simple video. Very simple illustration there, just copy and pasted. And there. So, and now here's a picture of cytosol containing all of the stuff, or just a cartoon image of cytosol. That's modeling what's inside. So here's a picture of cytosol showing microtubules, light blue. So light blue is going to be microtubules across there, or up here. And actin filaments, which is a dark blues. The dark blues actin filaments. And ribosomes, yellow and purple. So there's let's see, some yellow, purple. I believe this would be the yellow and purple is the ribosomes. And you also have soluble proteins, light blue, be a bunch of these ones there. And you also have, or it could be even this one, kinesin. Yeah, or actually I believe it's pronounced kinesin. So that's in red. Let's see, what's the red color? It could be these. Yeah, it could be this. That's the red one. And which is a protein that can move along the cytoplasm of animal cells. Protein that can move along and so on. And small molecules, which are white. And yeah, all these little dots right there. And RNA, which is pink there. The pink is the RNA. Yeah, so there's some RNA, and that's near the, it's RNA. I believe this would be it. Pink one there. This could be considered possibly pink as well. Actually, no, this is, probably this one, going through the ribosomes. And that's, so just double check that, but that's hard to see there. And now let's look at number 12, which is the lysosome, which is a membrane-bound spherical organelle that contains enzymes that can break down many kinds of molecules. So this is, again, another spherical organelle, or membrane-bound spherical organelle, and it contains enzymes this time to break down many molecules, many kinds of molecules or biomolecules. Let's go to number 12 in the overview. And where's the overview? Somewhere here. So number 12 is this big one here. It's bigger than this number 4 one. I think that was a vacuole or just a vesicle. Number 4 is just a vesicle, yeah. Number 4 is just a generic vesicle here, yeah. So now let's go back to number 12. All the way to number 12. So number 12 is right here, the lysosome. So here's a TEM transmission electron microscope views of various vesicular compartments. Lysosomes are denoted by LY. They are dyed dark blue due to their acidity. In the center of the top image, a Golgi apparatus can be seen distal from the cell membrane relative to the lysosomes. And let's just look at this. Here's a dark one here of LI, the lysosome. And this is a Golgi apparatus. That's because the shape is like that. And here's another view of this one. Here's the dark one right there. LY, and where's some other ones? Yeah, so the other ones, where could be? So this is LY. I'll have to zoom in to see where we could find one over here. I can't see one there, but this one here on the left side is an LY, and then in the top they are dyed dark blue. They're dyed dark. So in the center is the top image is the center, and this is distal from this cell membrane relative to the lysosome. So distal is the term distal, or the terms proximal from Latin proximus or nearest, and distal from Latin distar, to stand away from, are used to describe parts of a feature that are close to or distant from the main mass of the body, respectively. So you have something like this. So the hands are distal from the body, and then proximal is the shoulder over there, and so on there. So there's a different directional references over there. So the foot distal from the body, and the thigh there is proximal, and so on. So that is that one there. And let's just go back up here. So this one is distal. So a Golgi apparatus can be seen distal from the cell membrane relative to the lysosome. So this is so the cell membrane will be somewhere over here then, and it's further away from this than this one here. So it's further away than this. This will be proximal to the cell membrane. And fascinating stuff here. So this is X. It should be an LY somewhere there. But anyways, continue further. Yeah, so continue further here. So this is an anatomical directional reference. A caudal is from Latin caudum, or tail. So this is the caudal there, from Latin for tail. So it's going towards, and the cranial is upwards to the brain. And there's also the lateral ones. Medial is to the center, and lateral is away from center. And if you look at it from this angle, from the side view, anterior or ventral, and then you have posterior or dorsal, going to the back, dorsal fin, better. Yeah, dorsal fin and dolphins and sharks. So, and anatomy is from Greek anatomy, or dissection, is a branch of biology concerned with the study of the structure of organisms and their parts. Now let's look at number 13, which is the centriole, and this is a cylindrical organelle composed mainly of a protein called tubulin, and typically made up of 9 sets of short microtubule triplets. And here is the structure of the centrosome. We'll get to that in a bit, which contains these the centrioles, these two right here, and there is one that's called a mother centriole, and there is a daughter centriole over here, and I'll go over those in a bit. And then there is this paracentriole material, and there's these interconnecting fibers all across there, and these microtubules that shoot out from that. And let's just look at the overview image to see where this number 13 is. If we go all the way up, and I think we went too far up. So yes, it should be somewhere here. So number 13 is right here. There's the centrioles over there. Is the mother and the daughter one over there. So let's keep going further back to it. All right, so now we're back here. So this is the structure of a centrosome, and a centrosome, or the centrosome is Latin from centrum or center, plus Greek soma for a body, also called cytocenter, is an organelle that serves as the main microtubule organizing center, MTOC, of the animal cell, as well as a regulator of cell cycle progression. The centrosome provides structure for the cell. Centrosomes are composed of two centrioles arranged, these are mother and daughter, arranged at right angles to each other, or orthogonally, same thing as right angles. Yeah, or 90 degrees or perpendicular to each other, and surrounded by a dense, highly structured mass of protein termed the paracentrial material, the PCM. And this one right here, this microtubule organizing center, is a structure. So MTOC is a structure found in eukaryotic cells from which microtubules emerge. That's where they emerge from, that center location or structure. And now here is a mother and daughter centriole attached orthogonally or 90 degrees perpendicular. So this is the mother centriole, the way you can see the differences between it. Has distal appendages, so these are the distal appendages, these far ones. Distal means away from, let's say, assuming this is the center here. And then this is the subdistal appendages over here. It's subdistal, so not as far as this one. And there's the proximal ends, the close by ends over here. And there's interconnecting fibers on the bottom there. And these are microtubule triplets that come in triplets over here. And these are the distal ends, the far ends. And look at the difference here. This one has appendages on the mother one, and the daughter one does not have the appendages. So the daughter centriole. And so to explain further on these, it involves what happens during cell division. So before DNA replication, cells contain two centrioles, an older mother centriole and a younger daughter centriole. During cell division, a new centriole grows at the proximal end of both mother and daughter centrioles. Proximal is this close by end where they're close together there at this point. So another one grows there during cell division. After duplication, the two centriole pairs, the freshly assembled centriole is now a daughter centriole, and each pair will remain attached to each other orthogonally until mitosis. At that point, the mother and daughter centrioles separate dependently on an enzyme called separase, or separases. Or just separase, I think that's how I pronounce it. So the two centrioles in the centrosome are tied to one another. The mother centriole has radiating appendages at the distal end of its long axis and is attached to its daughter at the proximal end. Attached here, and then has these far away appendages on it. And each daughter cell formed after cell division will inherit one of these pairs. So we'll have either one or the other. And centrioles start duplicating when DNA replicates. And here's just a quick illustration of mitosis. I'll be going over this in depth later in this video. So mitosis divides the chromosomes in a cell nucleus. So here are the chromosomes here. First, you're going to have DNA replication, and then so they replicate, and then they separate. Now you're going to have this is the mitosis part where they separate. So, the DNA there duplicates, and then you separate the chromosomes. And then you get these two diploid cells, copies of each one there. Or two copies, or two sets of chromosomes on each one. And then going further, the DNA replication is the biological process of producing two identical replicas of DNA from one original DNA molecule. So if you have DNA right here, so what happens, they would split up, and then you have another strand come in and connect those identically to this one here. The DNA replication, the double helix is unzipped and unwound, then each separated strand in turquoise, the turquoise color right here, acts as a template for replicating a new partner strand, which is the green one. So the green one is a new partner strand coming in. So this green one, and then this turquoise it's the separated and acts as a template, and then it needs to connect with all the other letters. And the nucleotides or bases are matched to synthesize. Nucleotides, but the bases specifically are matched to synthesize the new partner strands into 2 new double helices, or helixes, or yeah, I'll just call it helices. Yeah, so you're going to get new bases connecting to this template strand over there. And briefly on the cell cycle, again, I'll be going over this later in this video in more depth. So the cell cycle or cell division cycle is a series of events that takes place in a cell that cause it to divide into 2 daughter cells. These events include the duplication of its DNA replication, and some of its organelles, and subsequently the partitioning of its cytoplasm and other components into 2 daughter cells in a process called cell division. Here's an illustration. So life cycle of a cell. And yeah, so here's how the cell cycle goes. And so you'll have a duplication process here. Actually, no, starting here. So you'll have chromosomes here duplicate, separate, and then yeah, and here's a zooming in of the DNA. I believe this is the separation point. And then yeah, then you have some structures that pull across it, pull them all apart, and then you have two these daughter cells like that. So yeah, this is just a quick illustration of it. And centrosomes are composed of 2 centrioles arranged at right angles to each other and surrounded. Okay, so we already have this part. Let's remove this. And here's an image of the centrosome. So centrosomes shown by arrow next to the nucleus. So there's the centrosome there, right next to this nucleus over there. I believe that's, or this irregularly shaped nucleus. Yeah, it's hard to tell what it is. But anyway, so that's a centrosome there. Let's see if there's a better image there. And here's an electron micrograph of a centriole from a mouse embryo. And here's those triplets, tubules there. There's triplets there. And this is a cross-section through it. That's very fascinating stuff. And now here is a 3D rendering of centrioles. And yeah, so here's its, I believe they would cut this out. And then they would zoom in. Here's a 3D rendering of it. So it would look something like this. So there's those 3 triplets across there in a staggered circle like this, in a spiral circle. So very interesting stuff. Yeah, so now let's look at number 14, which is the cell membrane. And yeah, this separates and protects the interior of the cell from the outside environment, also known as plasma membrane or cytoplasmic membrane. And let's go to the 14 over here on the overview. So the overview should be right here. And yeah, I think yeah, it doesn't include actually the 14 here. It just goes to 13. So 14 is just going to be around the structure of animal cell, depicting the organelles. Yeah, but the 14 doesn't show in here, but it is surrounding this cell. Let's go back to the 14. All right, so 14 is right here. And this is the structure of it. So illustration of a eukaryotic cell membrane. So there's a phospholipid, which has hydrophobic head and 2 hydrophilic head, which likes the water, and then you have the hydrophobic tail, or with 2 tails, that doesn't like the water, so it always moves away from that. And there's a phospholipid bilayer, and this is a zooming in of the cell membrane across here. And here's some various structures around it. You got carbohydrate sticking out there. You got some glycoprotein, which is a carb and protein mixture. You got this globular circular protein over here. Yes, and this one here is a protein channel or transport protein. And over here is cholesterol. And there's a glycolipid. This is a lipid and carbohydrate mixture, I believe. And then this one here is surface protein. This is surface because if this is zooming in onto this cell. So this is the bottom one. So this top part is the inside of the cell. The bottom is the outside. So that's the surface protein. There's another globular protein, integral globular protein. Yeah, and I believe it might be because it's on the outer surface there. And anyways, this is right here. Actually, no, this one here, integral. Yeah, that's what I mean. I know this is a surface protein. This one's an integral one. Yeah, it goes through the whole thing over there. And let's see, filaments of cytoskeleton. There's the filaments from inside. Oh no, this is from the outside cytoskeleton, which enhance the strength of that cell structure. And then over here, yeah, this was zooming in to get the phospholipid bilayer, and there's a phospholipid or phosphate dicholine. Let's try that again. Phosphate dicholine. Yeah, so anyways, this next one's alpha helix protein. Integral protein goes through across. and there's a peripheral protein on the outer edge over there. So he has a whole bunch of protein and molecules and so on. And there is the cell here. This is zooming in on this membrane part there. And there's a cytoplasm on the inside, and there's a nucleus over here, and there's the extracellular fluid, which is outside of the cell. Just fascinating setup over there. And here is another, these ones are just cartoon images. Here's a real image. A micrograph from a transmission electron micrograph, or microscope. Yeah, micrograph. Yeah, I believe this should be a microscope from a transmission electron microscope. Yeah, so I just put that SIC or SIC there to indicate, yes, it's exactly as copied and pasted. Showing a lipid vesicle. The two dark bands are the two leaflets or individual units of the phospholipids, akin to two leaflets of a leaf, comprising the bilayer. And similar images taken in the 1950s and 1960s confirmed the bilayer nature of the cell membranes. Very fascinating stuff there. And yeah, the leaflets, it's because the setup of this one here, you could consider these as kind of like leaves spreading out of this bilayer center part. And here's similar to this is a pinnate. This is a type of leaf, so a pinnate or feather-like arrangement, so it sticks out like that. So it's similar shape to the bilayer. Leaf of an acacia. Actually, it's pronounced acacia, I believe. I just Google search that. The pronunciation. So this is a type of plant with 10 leaflets there. There's 10. There's 5 on the side, 5 on the side. Yeah. And then, so here, yeah, so look at the outer leaf. So similar to how this setup is. Leaves are pushing outwards. So here's the outer leaflet, and there's the inner one over here. And yeah, so figure 4-3, I got this from this nap.edu website. So lipid bilayers in cells are asymmetric. In mammalians, asymmetric, so they're not symmetric. Yeah, in mammalian cells, the major lipids in the outer leaf are this right here. Yeah, this one I believe is pronounced sphingomyelin, or SM, the type of lipid. And then this one here, glycosphingolipid, GSL. And this other one, phosphodicloline. Or actually, let's try pronunciating these better. This one's glycosphingolipid, GSL, and this other one is phosphodidylcholine, or PC, and it's cholesterol, and then some. This is and some dioethanolamine, or PE. So this is this one is the outer leaflet. So there's the outer one. The inner one is where the cytosol is, or the cell membrane. It's outside of the cell. So this is the other one is cholesterol. There's GSL, there's SM, and there's PS, the different kinds of lipids. And the inner leaflets, major lipids are PE. So you also have this one right here, and then PS, which is phosphodilserine. Yes, or PS over here, and also this PI, which is phosphodiolinositol. Or actually, I think it's pronounced phosphodiolinositol, PI, and then this cholesterol, and some PC, which was this over here, phosphodilcholine. And yes, the source, London 2014, and the source is inside this link there. And reproduced with permission from Aaron Lundell, and yeah, this right here, exofacial solution. This means facing away from the cell. So exofacial, so facing, exo is away or apart, and so on, or without. So exo, yeah, so there's the facing away from the cell, and it's inside of the cells over here. And let's just zoom in right here, and there is the size of it, 5 nm. Remember, nanometers is billionth of a meter. So figure 2 is one that's from advanced-microscopy.utah.edu website. So a neuromuscular junction, or a type of synapse in S. elegans, which is a tiny 1 millimeter long transparent worm. The lipid bilayer of the plasma membrane is less than 5 nm, but the individual leaflets of the bilayer can be resolved. So you can still see a tiny bit of these little fragments there. And then photo is from this guy right here. And let's just try the pronunciation. So Shikaki Watanabe, and then Eric Jorgensen. Yeah, I just realized that I read this wrong, as I read this as S. elegans instead of what it actually is, which is C. elegans. So I'm not sure why I confused the C with an S. But anyways, continue further. So within the cytoplasm of a cell, there are many biomolecules such as proteins and nucleic acids. In addition to biomolecules, eukaryotic cells have specialized structures called organelles that have their own lipid bilayers or are spatially units. Basically take up volume and space, etc. These organelles include the cell nucleus, which contains a cell's genetic information, or mitochondria, which generates adenosine triphosphate ADP to power cellular processes. Other organelles such as endoplasmic reticulum and Golgi apparatus play a role in the synthesis and packaging of proteins, respectively. Biomolecules such as proteins can be engulfed by lysosomes, on another specialized organelle. And this was in regards to just a general animal cell. Now we'll look at plant cells. So plant cells have additional organelles that distinguish them from animal cells, such as a cell wall, chloroplasts, and a vacuole. Or vacuole. I think that's how you pronounce it. So now here's a overview schematic of a plant cell. So structure of a plant cell. So you have all of these parts. Yeah. So inside here, this is a vacuole, and inside, it's basically, we'll go over that in a bit. It basically has a giant blob there, or cavity for containing a bunch of stuff. So it usually has one giant large one, and there's a bunch of these other ones. There's a nucleus inside here. There's a chloroplast. There's a Golgi apparatus, similar to animal cells. There's cytoplasm. Is the everything inside, excluding the nucleus, and some other stuff here. Let's just continue further. So MES note, most mature plant cells have one large vacuole that typically occupies more than 30% of the cell's volume, and that can occupy as much as 80% of the volume for certain cell types and conditions. So yes, as a large vacuole like that. And here I quickly just teleported to number 10 of the animal cells one. Yeah, so this vacuole is just a membrane-bound organelle filled manually with water, containing inorganic and organic molecules such as enzymes and solution in certain cases, including solids which have been engulfed. So yes, these are these vacuoles, but in plant cells they usually have one very large one. So yes, and that make up from 20 or 30% to 80%. Let's just keep scrolling back to it. And yeah, so yeah, it's occupies more than 30% of the cell's volume, and can occupy as much as 80% for some cell types and conditions. The fluid in the vacuole is called the cell sap and serves as storage of materials and provides mechanical support to the cell. So it contains a bunch of stuff and also support for the cell, the structural support. The tonoblast is the vacuole membrane. So this is the vacuoles inside. There's a tonoblast around it. So that's the membrane around it. Let's continue further. So here is some vacuoles here. So this is the anthocyanin or storing vacuoles, which are just vacuoles containing water-soluble pigments, and this is for the color. See how it's purplish over there. This is Royo, or I think it's pronounced Rio Spathacia, and this is a spider wort, which is a type of wild flower. So yes, those are the cells inside there. The giant vacuoles inside the cells. So a pigment is a colored material that is completely or nearly insoluble in water, so it doesn't dissolve. In contrast, dyes are typically soluble, at least at some stage in their use. The difference between a dye and a pigment. This pigment doesn't mix in, but dyes do. So pigments for sale at a market stall in Goa, India. Or I think it's pronounced Goa, India. Let's continue further here. We can make this a bit smaller. All right, even a bit smaller there. We continue further, and here just to illustrate pigments, how they get the color. So a wide variety of wavelengths colors encounter pigment. This pigment absorbs red and green light, but reflects blue, creating the color blue. So yes, you have a pigment here that would absorb these wavelengths, these red and green right here, but then it reflects the blue one, doesn't get absorbed, and then hence you get this bluish color. Now, in this figure, what we have is sunlight encounters Roscoe R80, which is primary blue, or just quote primary blue pigment, and then the product of the source spectrums from sunlight, and it looks like this, indicating it has a lot of wavelengths. and the reflectance spectrum of the pigment results in the final spectrum and the appearance of blue. And yeah, so white light is how the eye perceives many wavelengths at once. So this would be white light. And yeah, so when you look at it like that, it's a bunch of wavelengths together, and then this is the primary blue reflectance spectrum, or these are the colors that, or the wavelengths that get reflected off this material. And then what you'll end up having is when you so you have these wavelengths that get reflected, and you have this input spectrum. You're gonna get a final one that looks pretty much similar to this. It's similar to the reflectance one, depending on just how intense the light you shine on it and other stuff like that. So it looks something like this, and then basically this one here has all of these is not reflected. So these ones are the absorbed. So the absorbed ones is over here. So this gets absorbed and then it becomes like this. And this one is blue light, which is blue is a higher frequency color over there. Let's continue further. So a cell wall. So plants, plant cells have. Some of them have this additional structure. So a cell wall is a structural layer surrounding some type of cells just outside the cell membrane. So if you have, here's a vacuole, here's a cell wall around it, and membrane would be inside, and then you have a cell wall around that. And there's a chloroplast. This figure is from, let's see where this figure is from. This is from schematic.org. Yes. Interesting stuff. There's a chloroplast and so on. We'll get to chloroplast in a bit. And that's a granum of a chloroplast. So plants are mainly multicellular organisms, predominantly photosynthetic eukaryotes of the kingdom Plantae. Here's just a taxonomical view of it. So you have life there, then you have a domain, you have a kingdom, then you have phylum, you have a class, order, family, genus, species. So plants is of the kingdom Plantae. So you have it over here, just below the domain and above the phylum. And yeah, so this figure, let's just read the description of this figure. So the hierarchy of biological classifications, 8 major taxonomic ranks, which is or just categorization ranks. A kingdom contains one or more phyla. Intermediate minor rankings are not shown. So anything in between and other minor ranks. And going further, so here's just the example, some further examples. This one's for a red fox. So the major ranks, domain, kingdom, phylum, class, order, family, genus, and species apply to the red fox. Volupus, or actually I think it's pronounced Vulpes, and then Vulpes. So the genus and the species, same name. And yeah, Vulpes, I had to Google the pronunciation. So yeah, so that looks like that. So then going above it, genus is Vulpes, family Canidae, or I think it's pronounced Canidae, Canidae, and then this is Carnivora, then Mammalia, then Chordata, this is backbones, and then Animalia, to distinguish from plants, and Eukaryota, so to have multicellular organisms. So yeah, so you can see how the domains goes up and up, and then there's life would be whatever is viewed as life, and etc. Yeah, relative to non-life, such as metals and so on. All right, let's look at the human one now, just for reference. So this is figure 3. So this one is from Wikipedia. This one is from, let's see this one. This is from biologyonline.com. And this is the taxonomy of humans, and source, you have Biology Online. And so you have domain is Eukaryota, so they're both multicellular organisms, and they're both animals. Red fox, and this is an animal, so Animalia. And then the phylum is Chordata, same as this, as a backbone. And then class Mammalia, so they're both mammals. And then this one is order Primates, so similar to monkeys and so on. And then the family Hominida. Actually, I think it's pronounced Hominidae. And then this is the genus is Homo, and then species Homo sapiens. And yeah, so you have adult human, male and female, Homo sapiens. Let's continue one more here. Another look at this. Yeah, that's usually how MES is. I gotta go in depth to see as much as we can find out. So here's figure. This one is from this website, BHAVANAGJAGAT.com. And this is Homo sapiens. Yeah, so we're going further, going backwards. Start from animals here, and this has a description here just to illustrate the difference. So you have Animals, organisms able to move on their own. That's the kingdom. Then you have the phylum, which is chordates, animals with a backbone. And then the class for humans is a mammal. Yeah, so it's a chordates with fur or hair and milk glands. And then the type of order is primates, mammals with collarbones and grasping fingers. And then it's family hominids, or you could use this more technical name hominidae. Yeah, hominidae. Yeah, so you could use that, or just go with these more general terms. So hominids, primates with relatively flat faces and three-dimensional vision. And then Homo, which is hominids with upright posture and large brains. And then Homo sapiens, members of the genus Homo, with a height forehead and thin skull bones. And yeah, I think this is a mistake I just realized. So it's not height forehead. Yes, it's high forehead. That should be a space. So let's just put this here. Yeah, here I just paused the video and just added it. So typo should be high forehead, not height forehead with a misspelling. Just fascinating stuff. That's a species, Homo sapiens. And now let's look at another view of the animal. I mean, the plant one. So yeah, plant is eukaryotic. Eukaryota. It's a multicellular organism. Then you have the kingdom Plantae, as opposed to the kingdom here, the animals or Animalia, where's the animal? Animalia for the kingdom. And then the next one is going to be the phylum is going to be Anthophyta. And then you have the class is going to be Magnoliopsidida. Actually, let's try that again. Now, this one here first is Anthophyta. Let's try it one more time. Anthophyta. Yeah, so Google pronounced that. And then this one, so it's a Google search. This one, so Magnoliopsida. Yeah, so Magnoliopsida. Then this, the order is going to be this one, Caryophyllus. Alright, actually I think it's pronounced Caryophyllales. And then next one, family, is going to be this one. Yeah, this is Drosoraceae. Yeah, I have to search that one again. I search that as well. Genus is going to be this, which is Dionea. And then the species, Dionea muscipula, or muscipilla. So Dionea muscipilla, and this is, this is this plant right here. And I have a whole bunch of terms there. And this one is from, this is carnivoresplantresource.com. It's very fascinating stuff. All right, so let's go to the next segment. So plants are photosynthetic plants. Photosynthetic organisms. And where was that? That's just over here. Let's just go read over on plants again. Yeah, right here. So plants are multicellular organisms, predominantly photosynthetic eukaryotes of the kingdom Plantae. Plantae. So it's photosynthetic, and photosynthesis is a process used by plants and other organisms to convert light energy into chemical energy that, through cellular respiration, which are reactions and processes in breaking down nutrients into ATP and then releasing waste products, we'll go over that later in the video, can later be realized to fuel the organism's metabolic activities. And chloroplasts are organelles that conduct photosynthesis, where the photosynthetic pigment chlorophyll captures energy from sunlight, converts it, and stores it in the energy storage molecules ATP and NADPH, while freeing oxygen from water in plant and algal. Or I think it's pronounced actually algal. And then there's an informal term for a diverse group of photosynthetic eukaryotic organisms that typically live underwater, so algal cells. And this right here, chloroplasts, let's look at the overview image of the chloroplast, and you can see the chloroplast is right here, these green ones over there. So there's a chloroplast over there, and there's some of its components inside. And also here's a mitochondria over here, or mitochondrion over here. And here is the cell wall, this one right here, and then around this yellow, that should be the cell membrane, which it is, you can see that right here, plasma membrane or cell membrane or lipid bilayer membrane, etc. Let's continue further here. So those are the chloroplasts, and here are some close-up images of it. And also there was one in this electron micrograph. There's the chloroplast right here next to the vacuole, and there's a cell wall. All right, continue further. So here is, so seen through a microscope, chlorophyll is concentrated within organisms in structures called chloroplasts, shown here grouped inside plant cells. So inside plant cells, you have a whole bunch of these, these green structures there, and those are the green chloroplasts. They're the chloroplasts, which conduct photosynthesis, and so on. So fascinating stuff here.